Greetings everyone, my name is Artur Queiroz, I'm from Fiocruz, Bahia, here in Brazil, and I would like to present my project that was a problem for funding in the CFAR. However, we haven't started yet, so instead I'm gonna present our work entitled Assessing Tuberculosis Risk in Pregnant Women from the PRACHITi study using transcriptomics. That is a work from our group that I shared called the first author with George from Cornell, and it was performed mostly because the TB diagnosis is quite a challenge. It's major performed by identifying the mycobacterium in the sputum for the patients. However, the accuracy is not quite good. So, and when we apply this approach in the patients with HIV, the accuracy drops too much, and to get a good diagnosis accuracy, we need more information such as cytometry, Luminex or X-ray, and all the clinical information from these patients, which allow us to identify which one of them are with TB or not. However, also the accuracy is not so higher than the previous ones, and recently they used the gene expression information trying to figure out how you can better diagnose these patients. Up to date we have almost 45 signatures, some of them addressing people living with HIV-1, but almost none addressing pregnant women or even pregnant women infected with TB and HIV. So we have helped increase this number of signatures when we analyze data from persons with advanced HIV-1. So these patients has a low CD4 count, and we use the data from different sites from India and also from Uganda, and we apply machine learning approach and identify two genes that were very good for TB classification in those patients. We also have used a multi-omic approach. We have used microRNA expression data with metabolomics and Luminex data from patients with advanced HIV, and we find that one metabolite, sorry, and one microRNA was also good for classification of these patients. In this work, we are trying to classify the TB infections in pregnant women with HIV. So we enrolled like almost 125 patients, some of them with TB and HIV, some of them only with HIV-1. And we could identify 7 pregnant women with TB and HIV and 28 with only HIV, and they have similar clinical and HIV status. So only in the pregnant woman with TB we have a higher proportion of first pregnancy compared to the control one. So there's this one difference between the groups that we find. And after that, we collected blood samples from these women, from the TB and non-TB women, and we followed them for the postpartum period. We also took this blood sample from these patients, and we extract the RNA to perform the gene expression analysis and the sequencing in the Illumina. So we have sequenced these samples, and I have used the quality control to remove all the bad reads, and also performed the alignment with Salmon, and we align these reads with the transcriptome of the human transcriptome reference. So after that, we got the chain count and performance the differential expression analysis with the SEC, and identify the DAGs, the different expressed genes, and we use them to investigate if we have any pathway that was enriched in any conditions. Aside this, we performed the decision tree approach, that is a machine learning approach that allows us to identify the minimal set of genes that was able to differentiate our condition, like the TB and non-TB woman. And also we get the same data from our patients and compare with the other previously published signatures. We performed the cross-validation of our genes that we find and compared to the other signatures, the performance, to see if they were better or worse than ours. So we are able to identify 469 genes in the comparison in the pregnancy, for comparing the pregnant woman with TB and HIV infected only with HIV-1, and the same comparison performance in the postpartum, and the similar number of genes were found with 424 genes. We took those genes and compared to the Reactome and the KEGG databases, and we just find pathways enriched in the postpartum differentially expressed genes. So the alpha deficiency was upregulated in the postpartum genes, while the translational of structural proteins and maturation of spike proteins were downregulated in this comparison. Which was quite surprising, because we were expecting like a neutrophil or interferon alpha or interferon gamma pathways, but we could not find nothing. And after that, the decision tree retrieved to us 2 genes that were considered the best model for classification in the pregnancy, and 2 genes from the postpartum comparison. So the GGT7 genes and the myom1 gene were classified as the best genes for the classification in the TB status in the pregnancy. So these women with TB had a lower GGT7 expression and also a higher myom1 expressions compared to the non-TB pregnant woman, while the TB women in the postpartum have a higher levels of ITSN1 and LONRF1 gene expression. So together, these genes have a very good accuracy, very good performance in the classification. Have like a area under the curve with 96 and 97, the both of the sets in its comparisons. But we compared them with the other ones. So also the cross validation showed that we have a very good accuracy in both genes, gene sets. Sorry, and with no information rate of 78 in the pregnant model, while it was 68 in the postpartum model. So the sensitivity was good in the pregnant model, however it's not so good in the postpartum model, while the specificity was different in the pregnant compared to the postpartum model. However, when you compare the other previous signatures, we can see here in the first comparison, that was the postpartum woman comparison, comparing which woman had TB and which one has not. So we have the best performance was the TB postpartum model, who was very good in this comparison, while the TB pregnancy model was good in the comparison in the pregnant woman. So our gene models outperformed all the previous ones, so it was nice. And we try to understand what is happening with our samples with MDP approach. It means that we took one group of baseline as a reference and took the median and standard deviation for all the samples and used this to perform a z-score normalization. And this means that this group is going to be the base of all comparisons between and across the groups, and we can measure how many variation one group has to the other ones using this as baseline. In this case, we use a healthy woman from all the cohorts that we have in our group. As I am responsible for all the analysis, I have this. It's easy to me to get this data and use them in different works. And when I done that, we use this woman as baseline and compare all of the conditions. So the pregnant controls, the woman with only HIV, and the pregnant TB, the woman with TB in the HIV, and the postpartum and postpartum TB. So we can see that we have more perturbation, more variation in the patients with TB in comparison to the controls, that is the people, this woman with only HIV-1. However, this was not significant, so we dropped this and used the other results. That was we have two strong and potential models for TB diagnosis in the pregnant women, even in the pregnancy and also in the postpartum. And it was interesting because we find the gene GGT7 is part of the glutathione pathway that is also associated with immune response against MTB. So this makes sense that we find this gene. Also the ATS1N1, this is associated with increases on the interferon gamma and TNF alpha in those patients. But the myome 1 and the long RF1, they were not associated with TB before that. So they these genes are source for new information, so we're gonna investigate more. But our work have some limitations. The first of them is the limited sample size. We have only 7 women in the case group, so we could not find more pregnant women with TB and HIV. So, and we needed to validate in the other cohorts, because in the previous work that we find, we compared sample for Kenya, Malawi, and South Africa. We could see that the performance of the signatures vary too much between the places, and also we can see here is a comparison of children from Malawi and South Africa. They have different performance when you compare the adults from the both sides. And also in the Kenya we have patients with culture positive confirmations. They were superior, the performance was superior from the culture negative. So the accuracy of signatures could vary, but we're not sure why. So we should test in more data. I looked for the literature for available data from pregnant women, but I could not find any with TB and HIV to increase our sample size. So I would like to thank the Department of Pediatrics and the Gynecology from BG Hospital, because without them we could not make the work. They helped a lot with the patient management and also take care of these patients. Also the persistent system from Pune, India, that will help us in the data transferring. We'd like to thank the funding from Eunice Schieber National Institute, the National Institute of Allergy and Infectious Diseases, the GDR Foundation, the McHa Foundation, and Wincounter Foundation. Thank you for watching me.